My name is Nina Bauer. I'm the Executive Vice President for Strategy and Commercial at FUJIFILM Cellular Dynamics. New approach methodologies are also called new alternative methodologies by some people. They essentially enable drug developers to avoid using animals. So when you're in university, you're usually taught the 3 R's: replace, reduce, refine. To really be conscious about using animals in research. New approach methodologies go one step further. They're really trying to bring in new technologies and leveraging those to really get away from animal models. So that includes in silico technologies, which leverages AI and computer modeling. That includes high-throughput screening with in vitro assays. That also includes microphysiological systems, which is organoids, organ-on-a-chip type approaches. So they all try and replicate human tissue, both healthy and disease, in a much more relatable and relevant way than any animal ever could. In the EU in 2010, there's a directive that encouraged the adoption of 3 Rs, and that is expected to go into law this year. It was expected to go into law last year, but that hasn't yet happened. In the US, the FDA Modernization Act 3.0 was put into law last year, which comes with a little bit of funding as well. So there's government push to get companies to adopt more alternative approaches to drug screening, to toxicology testing, to release testing, things like that. And so with that government push, now it has entered mainstream media, it has entered really funding agencies. So the NIH now will actually, if you have an animal model, in some cases we heard the NIH will say, why don't you move into a NAMS approach rather than this mouse model? So government push, but also if you're adopting NAMS, it has a cost benefit, it has a time benefit, it's faster, it's cheaper. You have a model that is really human relevant, because it is often the cellular models are based on iPSCs, which is our expertise and our strength. And then you can do much more high throughput screening using computer models and things like that in a much more abbreviated time frame. All of these new technologies enable replacement of animal models. And we recently discussed that, and there will be a gradual move or shift. It will not be tomorrow there's only going to be NAMS. But I think companies are really starting to see the benefits of moving into these systems. It's much cheaper. If you develop an animal model, you have to go through certain breeding cycles, and you won't know until an animal is born whether the edit or the model has actually worked. Whereas in a cellular system, the throughput is much higher, and you can much more quickly get your results and whether you have a model that works. It has human relevance, and you can use cell lines that have actual human disease genetics that you can then turn into an in vitro system, for example. The high throughput also allows to really using AI models and machine learning to adopt mechanistic or identify mechanistic context that an animal would never tell you. And so overall, these new technologies, I think, will be really beneficial for drug development and just being much more cost effective. And quite honestly, I've been to so many conferences that I attended 20 years ago as a student, and I still see the same posters, talks on the same topics. We're still asking the same questions. And I think the model so far that we have used just has not developed. And I think now we're at this juncture where we can really find new approaches and ask questions very differently, and hopefully that will really generate new findings and ultimately lead to the development of new drugs.